This small pharmaceutical vial is far more than a container. It is a promise of health. Its creation is governed by one of the most stringent sets of standards in the world: Current Good Manufacturing Practices, or cGMP. The process isn't just about filling a bottle. It's about absolute, unwavering control over every variable, ensuring patient safety is never compromised. We are tracing the full, 20-minute journey of the vial, from raw glass to final patient release. The preparation stage is paramount. We begin with the three critical components: the vial, the stopper, and the sterile drug product. The glass vials, usually type 1 borosilicate, are carefully engineered for strength and chemical inertness. They arrive at the facility and are immediately subjected to rigorous washing. This isn't just tap water. We use water for injection, or WFI. WFI is purified through a combination of techniques like distillation and reverse osmosis to remove virtually all ions and impurities. The quality of this water is continuously monitored using sensors that check for conductivity and total organic carbon, or TOC, ensuring its purity is beyond reproach. Following the wash, the vials enter the depyrogenation tunnel. This machine is split into three zones: the infeed, the high heat zone, and the cooling zone. In the high heat zone, often reaching temperatures above 300 degrees Celsius, the dry heat destroys the stubborn endotoxins. The temperature gradient must be meticulously controlled to ensure maximum effectiveness while preventing thermal shock to the glass. Importantly, sterile, filtered air moves through the tunnel in a laminar flow pattern, preventing any non-sterile room air from entering the clean zone. Simultaneously, the stoppers and caps are undergoing their own processing. Stoppers, typically made of bromobutyl rubber, are treated for several hours in specialized washing equipment to remove any manufacturing residue. Meanwhile, the drug product itself is being prepared in the compounding area. This is where the active pharmaceutical ingredient, or API, is carefully weighed alongside excipients, stabilizers, buffers, and preservatives, using validated, high-precision scales. The mixture is dissolved, usually in WFI, and then agitated until homogeneous. Before it is pumped to the filling suite, the solution is forced through a 0.22 micron sterilizing filter. This absolute filtration step is the final physical barrier against microbial contamination. The pressure and flow rates across this filter are carefully monitored to ensure its integrity is maintained throughout the entire batch. Now, we enter the most highly regulated part of the process: the aseptic core. This environment is classified based on strict ISO and cGMP standards. The filling machines operate in a Grade A zone, often under a specialized barrier system like an RABS or an isolator, which provides a physical and aerodynamic separation from the surrounding Grade B or C cleanroom. The sterility of this environment is constantly verified through environmental monitoring, or EM. Air samplers draw in a measured volume of air onto a growth media plate, while contact plates are pressed onto surfaces like floors and equipment. Crucially, personnel are monitored using glove prints, taken at key intervals. If any of these monitoring points show contamination above strict limits, the entire process is stopped, and the batch is at risk. The sterile, cooled vials now enter the Grade A environment and queue up for filling. The filling equipment is selected based on the nature of the drug. For high-volume, long-run liquids, high-precision piston pumps, which offer exceptional speed and accuracy, are often used. For viscous products or when dealing with highly expensive biologics, peristaltic pumps are preferred. These pumps use tubing that is disposable and single-use, drastically reducing the cleaning validation burden and minimizing cross-contamination risk. The core principle of the line is no vial, no fill, governed by photoelectric sensors. The pumps dispense the exact programmed volume, and the filling needles retract as the vials move forward. Throughout this run, statistical checks, known as in-process controls, are continuously performed. Operators verify the fill weight against the target, often every 15 to 30 minutes, ensuring the pump calibration has not drifted. Immediately following the fill, the vial passes under the stoppering head. Rubber stoppers, fed from a sterile vibratory bowl, are carefully picked and placed into the vial opening. The speed here is critical. The vial must be sealed as quickly as possible under the laminar airflow to minimize the exposure time of the sterile drug product. This is the point where the process diverges. For standard liquid products, the stopper is fully seated, and the vial moves toward capping. However, if the drug requires freeze-drying or lyophilization, the stopper is only partially seated, often called a gapper. These vials are placed onto pre-chilled stainless steel shelves and loaded into the massive lyophilizer chamber. All vials, whether liquid or lyophilized, then proceed to the capping station. An aluminum overseal is placed over the stopper, and a set of rotating crimping jaws rolls and deforms the aluminum skirt tightly around the glass lip. This crimp is vital for maintaining container closure integrity, or CCI. Validation of this process involves physically measuring the residual seal force and the height and width of the crimp to ensure the seal is robust and consistent. A failure here means product instability and a potential path for microbial ingress. The moment of truth follows: inspection. Since sterility cannot be retested without destroying the product, the only way to confirm a defect-free product is through meticulous visual inspection. While human inspectors, working under controlled lighting known as a light booth, remain essential for training and complex or low-volume products, most vials now pass through automated visual inspection, or AVI. AVI machines use a combination of high-speed cameras, stroboscopic lighting, and advanced image analysis algorithms. They spin the vial to introduce controlled fluid motion, allowing them to detect particles floating in the liquid. They check for fill level, glass cracks, cosmetic flaws, and proper cap crimping. The system captures and processes dozens of images per vial in seconds, automatically rejecting any defective unit. Even after 100% inspection, a final, statistical quality check known as AQL, or acceptable quality limit, sampling is performed on the accepted batch. A small, statistically relevant quantity of vials is pulled for final destructive testing. This destructive testing is the final scientific confirmation. The sterility test involves transferring product samples into a growth media and incubating them for 2 weeks to confirm no bacteria or fungi are present. The LAL test, Limulus amoebocyte lysate, confirms the absence of endotoxins. Furthermore, assay and potency tests verify that the drug concentration remains within specification. All of these processes, the compounding, the washing, the filling, the M-data, the inspection, and the final lab results, are documented in a comprehensive batch record. This document can run to hundreds of pages. It is meticulously reviewed by the Quality Assurance, or QA, department. Every signature, every parameter reading, and every deviation, no matter how small, must be accounted for and justified. Only when the QA team is satisfied that the entire manufacturing process adhered strictly to cGMP, and the laboratory results confirm the product's quality, is the final signature applied, the QA release. The vials, now fully released, move to final packaging. They receive their multi-panel labels and are packed into cartons along with mandatory patient information leaflets. The final and increasingly critical step is serialization. Each carton is marked with a unique, encrypted 2D matrix code that can be tracked across the global supply chain, serving as a powerful defense against counterfeiting and ensuring that only verified medicine reaches the patient. The finished cartons are boxed, palletized, and moved into the warehouse, often a cold chain environment requiring specialized refrigeration and temperature mapping. The journey is complete. The product has been manufactured, documented, tested, and released. The pharmaceutical vial is a masterpiece of complex engineering and relentless quality control. It is a testament to the fact that in drug manufacturing, perfection is not a goal. It is the absolute minimum standard. From a glass tube to a finished, authenticated therapeutic dose, this intricate process guarantees safety, integrity, and trust, ultimately connecting the innovation of the scientist to the health of the patient.